This video is about structure and functions of cell membrane. Cell membrane is a general term refer to all the biological membrane found inside and enclosing the eukaryotic cells. All cells covered by cell membrane. Eukaryotic cells have membrane-bounded organelles. So the plasma membrane and the organelle membrane have the same structure. In 1972, Singer and Nicholson proposed the cell membrane as Fluid Mosaic Model. They proposed that cell membrane consists of a bilayer of phospholipids with various protein molecules embedded and attached to it. So they say that cell membrane is selectively permeable, meaning that it only allows some certain molecules to pass through, not the others. So this is the Fluid Mosaic Model proposed by Nicholson and Singer. It consists of the phospholipid bilayer and the protein embedded with it. So Fluid Mosaic Model has two components, major component and other component. The major component composed of phospholipids and proteins, while other components such as carbohydrate and cholesterol. So this is the structure of Fluid Mosaic Model, where there is protein, cholesterol, glycolipid, carbohydrate, glycoprotein. So according to this model, it is known as mosaic because the proteins are embedded in the phospholipid bilayer. Fluid because proteins and phospholipids are free to move. They are not static. But the movement is only from side to side, which is lateral movement or flip-flop movement. However, it is not through the membrane. Okay, so this is the movement side to side, lateral movement. Flip-flop means from top to the bottom or in other way. So there are some factors which influence the fluidity of the plasma membrane. Okay, membranes are always fluid. They are not static. So membrane held together by hydrophobic interaction, which is much weaker than a covalent bond. So membrane fluidity can be affected by three factors. Factor number 1, movement of phospholipid and some proteins. Factor number 2, phospholipids with unsaturated hydrocarbon tails. And number 3, increase in temperature increase the fluidity of the membrane. So let's look at the first factor. So the movement of the phospholipid and some proteins. So as we say that the phospholipids are not static, they always moving. So the movement is either laterally or flip-flop. So this movement makes the membrane fluid. Second factor, phospholipids with unsaturated hydrocarbon tails. So when the hydrocarbon tail is unsaturated, they form the kink. So this kink prevents the packing of the phospholipid bilayer together. So this keeps the molecules from packing together, which enhance the membrane fluidity. So imagine if all the hydrophobic tails are saturated, all of the phospholipid, they will be closely packed together, which makes the membrane hardly move. So for this membrane, it is known as viscous, which is more static. Compared to hydrocarbon tail, which has the kink, it is more fluid. Third factor, increase in temperature increase the fluidity of the membrane. So there is cholesterol found in between the tails of the phospholipid bilayer. When the temperature is moderately warm, the cholesterol will reduce the membrane fluidity by preventing the free movement of phospholipid bilayer. Because when temperature increases, the phospholipid will tend to move more vigorously. Hence, cholesterol here help to prevent the vigorous movement of the phospholipid. On the other hand, when the temperature drops, cholesterol molecule will prevent the tails of the phospholipid bilayer from joining together and slow down the solidification of the membrane. So when the temperature starts to cool down, cholesterol here prevent the phospholipid bilayer from joining together. Because when the phospholipid bilayer join together, the membrane tends to become solid. Solidification may result in the permeability changes and enzyme deactivation. It is more harmful to the cell. Okay, so these are the cholesterol molecule which found in between the phospholipid. So for the selectively permeable, the movement of substance in and out of the cell, the movement of the molecule can be through simple diffusion for some small molecules, example oxygen, carbon dioxide, and hydrophobic molecule. Because of the phospholipid bilayer is hydrophobic, so they only allow some small molecule to move through the membrane down the concentration gradient. So example, some small molecules here, they can move through the phospholipid bilayer by simple diffusion. However, there are some large molecule or ions or polar molecules, okay, which cannot move through the phospholipid bilayer easily. So they have to move through some channel proteins. So these channel proteins are embedded in the cell membrane and have a pore for the materials to cross. Or there are some carrier proteins which can change their shape to move the material from one side to another side. Active transport which require energy to move the ions. Example, we have sodium-potassium pump. We also have other types of active transport such as endocytosis and exocytosis will move through the cell membrane. Okay, so the first one, simple diffusion, only move across the phospholipid bilayer. Second type is facilitated diffusion, which need the help of the channel protein, while the third type is the active transport, which need the carrier protein that need energy. Okay, different types of the molecules move through different types of transportation. So components of the plasma membrane, phospholipid bilayer, proteins, carbohydrate, and cholesterol. So we look at the first structure, phospholipid bilayer. So the membrane is about 8 nanometer thick. It consists of the phospholipid bilayer where the polar head facing outward, non-polar or hydrophobic tail facing inward towards the middle of the bilayer. So the function of phospholipid bilayer is to form the boundary to isolate the cell content from environment, also restrict passage of hydrophilic substance across the membrane. So the head facing outward while the tail facing inward. It causes inside of the membrane or the phospholipid bilayer become non-polar or hydrophobic zone. So when we open the cell like this, we can see that the head is facing outward and inward while the tail is in the middle of the phospholipid. Second structure which can be found in the plasma membrane is the globular protein, which form the mosaic part of the Fluid Mosaic Model. So we have two types of the protein. Integral protein, also known as intrinsic protein. So intrinsic means it is inside, embedded partially or fully inside the membrane. Second kind of protein is peripheral protein, also known as extrinsic. The meaning of peripheral means at the side. So extrinsic is outside, outer or inner surface of the membrane. Okay, so this is the integral protein, intrinsic protein. This is extrinsic protein, which is at the outside of the membrane. So phospholipid is amphipathic, means both the hydrophobic region and hydrophilic region. So hydrophilic, they like water. So when they're exposed to the water can easily dissolve and move across. Hydrophobic means they do not like water. Okay, so the center part of the membrane is hydrophobic region, which do not like water, while the outer part and the inner part is known as hydrophilic region. So there are some functions of the protein. First function as a transport because they can provide a hydrophilic channel. Okay, since water cannot move through the membrane easily because of the hydrophobic region in the center, so it provides a channel for the protein or other hydrophilic substance to move across. Second function, act as the enzymatic activity. So some of the protein act as the enzyme. Example, ATP synthase, which help to produce ATP. Function is signal transduction involved for the hormone. So they act as a chemical messenger, which help to send some chemical message to the cell. Intercellular joining, they join cell to cell by using the protein. Cell recognition, recognize which cell is the self cell, which cell is not the self cell, because they serve as identification tag. With the identification tag, the cell can easily recognize which cell belong to the same organism or the self cell. So when there is a non-self cell enter the body, it will be attacked by the antigen of the immune system. Last function of the protein is attachment to the cytoskeleton and ECM, which is extracellular matrix. Carbohydrate, either they bind to the protein, they known as glycoprotein, or they bind to lipid, known as glycolipid. So glycoprotein, the function is cell-to-cell recognition, protection, immune response, reproduction, and also cell adhesion. While glycolipid function also for cell-to-cell recognition and to provide stability for the cell and help the cell to join to other cells to form tissue. Cholesterol, located between the phospholipid layer, between the phospholipid molecule in the plasma membrane. So the function of the cholesterol help to regulate membrane fluidity and help the cell to maintain optimal level of fluidity.